Hi, everyone, and welcome to Night School. I'm Lynn.
And I'm Aria.
And we're part of the Nightlife Night School team at Cal Academy in San Francisco. We mix a little science, music, culture, and more, bringing this to you both in person at our weekly Thursday night Nightlife events, which is where I am right now, and virtually on our monthly Night School programs. Just a note, Night School is on the first Thursdays of every month, so put that in your calendar and tune in. Tonight, we're talking about the human and ecosystem relationship and if our relationship with nature can foster human health.
Yeah, so like Lynn said, we're talking just about humans and nature and how we're all interwoven in our different ways from, big to small, like lots of different scales. And to do that, we've got a really stacked lineup today. So I'm going to give like a really brief overview of everybody. Because they're way more interesting than me. So first we have Doctors Jack Dumbacher, Dan Miner, and Fayal Abderemane Ali, and they're going to talk about their research about poisonous birds and frogs and the toxins within them and how that can lead to a bunch of medical implications and medical cures, potentially. Then we're going to hear from Dr. Shannon Bennett, who's a virologist, on the origins of infectious diseases and how those can be intertwined with human health and what that may mean for future pandemics. And finally, we're going to hear from Dr. Jessica Hernandez on the critical importance of incorporating Indigenous knowledge into the fight against climate change. And she's also going to share a little bit about her upcoming book called Fresh Banana Leaves, Healing Indigenous Landscapes Through Indigenous Science. And as usual, full bios for all of these amazing folks are in the description below if you want to check those out and see some links. But yeah, pass it back to Lynn.
As always, tonight's program is live, so make sure to say hi in the comments. I know people have been already listing where they're from, so continue to do so. It's one of our favorite things to see. Also, shout out to anyone watching in the building right now at Science Today. Please also let us know if it's your first time joining or if you've been a Night School regular. We'll have Q&As after everyone's presentation, so make sure to put your questions in the chat. And with that, we are going to turn it over to Jack, Dan, and Fayal.
I'm Jack Dumbacher. I'm curator of birds and mammals here at the California Academy of Sciences, and I'm here with two of my colleagues, Dan and Fayal, and I'll let them introduce themselves.
So, hi, I'm Dan Miner. I'm a professor of biochemistry and biophysics at UCSF in the Cardiovascular Research Institute.
Hi everyone, I'm Fayal Abderemane Ali. I'm a postdoc in the Cardiovascular Research Institute at UCSF.
So great, and it's great to have you guys here with us tonight. We're going to talk about a paper that we just published in the Journal of General Physiology that we're very excited about, some work that we've been doing over the last couple of years. Wonder if you can cue up our slide presentation there, Lynn. And we're going to be talking about auto-resistance in poisonous pituhui birds and poison dart frogs. I first got started in this work many, many years ago when I was handling this bird, the hooded pituhui from New Guinea. And when you handle it in the field, it can easily cut your hand. And you know, we were just taking them out of the nets because we were studying birds. And as we let them go, we'd get these little cuts. You'd lick your fingers like this, and then your mouth would begin to tingle and burn and go numb. And after this happened to us a couple of times, we realized there was something weird about these birds, and we asked the local folks about them, and they said, oh yeah, you shouldn't touch those birds. They're poisonous. And so we became really excited about this, and we started researching them because we had never heard of a poisonous bird before. And just to give you an idea, this is how big the bird is next to me. So it's about the size of a jay here in the Western United States, and they can easily cut your hands if you're holding them. And so we started to begin to ask all those different questions that you ask when you have a poisonous bird. And the first one is, well, what poison does it carry? And so this took us a couple of years of work, but we were able to isolate and identify this toxin. It's known as batrachotoxin, and it's a steroidal alkaloid neurotoxin. So what the heck does that mean? So the first part is that it's a steroid. So it has this four-ring sort of foundation that all steroids have, like cholesterol, testosterone. And so that makes it a steroid class. It's an alkaloid because it has a basic nitrogen that's here in this ring that gives it certain chemical properties that allows us to isolate it and identify it as an alkaloid. The last thing is it's a neurotoxin. It's a neurotoxin because it binds to voltage-gated sodium channels in nerve and muscle membranes. And Dan and Fayal are going to talk a lot more about sodium channels tonight. But for now, those are the little channels that open and close and regulate the flow of sodium across the cell membrane, and that's what's responsible for that electric pulse down a nerve or across a muscle membrane. And so the batrachotoxin shuts that down, and because of that, it's one of the most toxic natural substances known, and because that channel is found in basically every animal that has a central nervous system, it's expected to affect a very wide range of target species. Now, batracho is Greek for frog. So batrachotoxin means frog poison. And so these frogs were previously known from this genus of frogs, the Phyllobates poison dart frogs. Now, of all the so-called poison dart frogs, only 3 species were ever actually used by the Choco and Embera Indians of Colombia to poison dart tips. And they would use these really cool blowgun darts. And they would use Phyllobates aretania, Phyllobates bicolor, and Phyllobates terribilis to do this. And terribilis is so poisonous that just holding it in your hand is dangerous and can actually kill you. So once we knew what the toxin was, we were interested in how the toxin is spread around the birds. We were interested in the evolution of toxin. We did all kinds of studies for many, many years. But today we're going to talk a little bit about auto resistance and why do the birds not get poisoned. So one of the key pieces of data that we had is this graph where we looked at which tissues contain the toxins. And we did find, in fact, I should say that here on the y-axis, this is micrograms of batrachotoxin equivalents per gram of tissue. That's just a lot of fancy words that say how toxic is this tissue, right? These are some of the different tissues that we looked at: skin, feathers, heart, liver, and muscle. And so even though the tissues on the outside of the bird and skin and feathers are the most toxic, and these are the ones that a predator or an enemy of a pituhui is going to encounter first, we still found that there was quite a bit of toxin in the heart, liver, and muscle. And these are the tissues that have a lot of those voltage-gated sodium channels. And it might look at first like, oh, well, but there's not very much, right? Not very much compared to skin or feathers. But just to put this into perspective, 0.05 micrograms of batrachotoxin, if injected subcutaneously into a mouse, will kill it within a minute. And so that means that every gram of muscle in a 65-gram bird has 100 times that. So that means that each gram of muscle has enough to kill 100 mice. And so this suggests that somehow the birds have enough of the toxin in their bodies. So it's not just that they're putting it on their feathers to make themselves toxic. They actually have it inside their bodies. And the question is, why do they not poison themselves? And that's the critical question that we were interested in. Now, in general, how do animals avoid auto-intoxication? There's 3 basic mechanisms. The first one is that you can enhance the ability to detoxify or eliminate toxins. That's what we do when we drink too much alcohol. Our livers break it down and our kidneys get rid of it, right? But that's not what's happening in the pituhuis, because they're clearly accumulating the toxin and it accumulates throughout their entire bodies. So the next 2 mechanisms are what we're going to be talking about tonight. And in fact, the mutations in target proteins is one of the things that most people in our field have been looking at. And there've been many, many published papers about these different target proteins and mutations that render them resistant. So I'll pass it on to Dan.
Okay, thanks, Jack. So I'm just going to talk for a bit about sodium channels. So as Jack mentioned, your nerves and muscles run on bioelectrical signals, and these are generated by proteins that live in the membranes that surround all cells. And what they do here is demonstrated on the left-hand part of the slide. So there's ions in the outside world, there's some ions in the inside world, and these proteins, which are called ion channels, sit in the membrane, and usually they're closed. And if you just advance for a second, when a signal comes, they open, and there's a flood of ions that come into the cell, and this causes the membrane potential to change and to generate an electrical signal. And a key thing about these ion channels is that they have to move very quickly from this closed to open state to allow the signal to happen, but then they also go back to something that's like a closed state, so the signal stops. And that whole process is essential for making sure that there's high fidelity transmission of electrical signals in your nerves. Now, as Jack mentioned, the target of batrachotoxin is a class of channels called voltage-gated sodium channels. And if you show the next part of the slide, these are a specialized type of ion channel that live in cell membranes, and they start this electrical signal. We know a lot about their structure now. We also know that there are a lot of toxins that target these channels because they're so essential to the function of the nerves and muscles. And you can see some of those diagrammed here. Many of these work from the outside. So these toxins on the outside are protein toxins that are made by various poisonous organisms, like scorpions, for example. There's also tetrodotoxin, which is a famous toxin found in fugu, which is the Japanese pufferfish, that can kill you, but if there's just a little bit in the fish, it actually gives you a very nice sort of tingling sensation in your mouth if there's a good chef. And these channels are also the target for things like general anesthetics. When you go to the dentist and they numb your gums so that they can work on your teeth, they actually are injecting a molecule called lidocaine that blocks sodium channels and temporarily stops the signals. And this is where batrachotoxin comes in. It's actually kind of a funny toxin because instead of stopping the channel by blocking it, it actually keeps it open like a doorstop. And so this process of going from closed to open and back to closed is broken, and there's just a constant flood of ions into the cells. And this is why basically the nervous system stops working, and that's why this toxin is so dangerous. And now I'm going to pass it over to Fayal, who's done some beautiful experiments to try to understand the origins of how the poison birds and poison frogs avoid this problem, because remember, these guys are both full of toxins. So it's good for them to deter predators, but they also have to figure out some way to not kill themselves. Fayal?
Okay, thanks Dan, and thanks Jack for the nice introduction. So now you all know about batrachotoxin effect on the sodium channel. So here I'm showing an illustration on how this toxin affects the channel and the kind of data we can see in the lab. And on the left, you have sodium channel activity where the channel, it can be open and then it can immediately be closed. But now on the right side, you can see right now that when the batrachotoxin is present, the channel can open but can no longer close. And that's problematic, as Dan mentioned. This is an illustration on a human sodium channel. So the experiment we did was to isolate the sodium channel from the pidihui bird and the terribilis frogs, 2 species that carry batrachotoxin, and test their sensitivity to this toxin. So in the next recording, you can see now this is the activity of sodium channels from the pidihui bird, and on the left, but on the right, you see that when we add batrachotoxin, the channel behaves just like the human sodium channels. It can no longer close. And this is the first surprise of our experiment, to show that this channel that is found in a bird that carries batrachotoxin is highly sensitive to that toxin. And on the next piece of data, we did the same experiment on the terribilis frog, where you can see again that in presence of the batrachotoxin, the channel can no longer close. So this also shows that this channel found in this frog that carries batrachotoxin is highly sensitive to this toxin. So this is the first experiment we did. We're very surprised by this, but this shows that mutation, the channel mutation, is not what protects these animals against batrachotoxin. And next, we did experiment where we challenged the animals. So we used 2 different kind of frogs, challenging them with batrachotoxin. And the next, you can see that when we did the challenge experiment on the Xenopus laevis, which is a non-poisonous frog, on the black trace here the experiment consists basically in anesthetize the frog and then inject them with either PBS, which is a control buffer, or the toxin, batrachotoxin. So the black trace is injection with PBS, and the pink magenta trace is injection with BTX, batrachotoxin. And you can see clearly that there is a big difference, that with when this frog is injected with batrachotoxin, the frog wakes up earlier and it ends up dying, basically. So it does not survive the toxin. And now when we did the same experiment on a poisonous frog, Phyllobates terribilis, here you can see that both the black and magenta traces are basically the same, which means that this frog is resistant to the toxin and can easily survive this toxin. So this experiment taught us one thing very fundamental, is that here we have a frog that has sodium channels that are sensitive to the toxin, but the animal itself is very resistant to the toxin, which means that there is something that shields basically these sodium channels from seeing the toxins in the first place. And the next experiment we did was also to challenge these frogs with 2 other toxins, saxitoxins and tetrodotoxin, STX and TTX, as Dan mentioned earlier. And here you can see that again, the non-poisonous frog, Xenopus laevis, is very sensitive to these 2 toxins because they cannot survive these toxins. But on the right, you can see that Phyllobates terribilis can survive after injection of these 2 toxins, which means that, and by the way, this the sodium channels from this Phyllobates terribilis are sensitive to these 2 toxins. So again, here we have something shielding the sodium channels from seeing these toxins in the frog. And the hypothesis was that there is a sequestration mechanisms that can basically capture the toxin and prevent it from reaching the target. So to test that, we did the next experiment where we used a poison frog. We used a saxophone, which is a protein that has been studied in the lab since several years ago. So Dan, if you want to talk a little bit about this saxophone before I present the experiment.
Sure, Fayal. So saxophone is a protein that was found in the American bullfrog, and it has this very funny property. So it binds this toxin, saxitoxin, that you see here in the picture, which is actually a very interesting toxin. It's made by algae, microorganisms that make red tide. It's actually identified here in California by Herman Summers in the 30s, working at UCSF, as the causative agent for something called paralytic shellfish poisoning. So for those of you who go to the coast, you may know that you'll see signs basically from May to October that tells you not to collect shellfish. And the reason is because it could be contaminated with saxitoxin, which is a super toxic agent, actually so toxic that it's listed as a chemical warfare agent. And Herman Summers, a long time ago, noticed that frogs were actually pretty resistant to this toxin. And in the 80s, this protein called saxophone, which is a soluble protein that floats around in the plasma and the blood of the frogs, was identified as a high affinity binder of saxitoxin. And a few years ago, my lab determined the structure of this protein. You can see it here on the left. Saxitoxin is bound there, really like a catcher's mitt binds a ball. There's a pocket in which saxitoxin fits perfectly. And we wanted to test then whether this kind of protein, it's the only candidate protein that's been well characterized that could be acting like a toxin sponge and maybe acting as a protector of the frogs from various toxins. And so Fayal can explain the experiment he did to test this in a sort of proof of concept experiment, to just ask the question, we know that the toxin binds very tightly to the channel. We know that the toxin can bind very tightly to saxophilin. If you poison the channels and then come in with saxophilin, can you rescue them? Is that sufficient? Yeah. So Fayal, I'll let you.
That's right.
The experiment there. Yeah.
So the experiment here on the right is we're going to look at these three traces. So the one on the left is so the differences between the three are the amount of saxophilin added in the experiment. So the one on the left, we added only 0.1 to 1. It's a molar ratio of saxophilin. So here the black trace is the control of the channel activity, and then when we added saxitoxin, that's the red trace, we can block the channel. Now, if on top of that we bring saxophilin, we can see whether saxophilin can rescue the channel activity or not. And here, that's the green trace, where you can see that now on the middle trace, when we have a 1-to-1 molar ratio, we rescue 50% of the channel. And on the right trace, you see that when we have 2-to-1 saxophilin saxitoxin molar ratio, now we can rescue 100% of the channel activity. So the presence of saxitoxin is not affecting the channel activity when saxophilin is present. And on the next graph here, this is just to give you an idea how fast this recovery happens. So basically, when the channel is open, then we apply saxitoxin. The channel is completely blocked, but when we add saxophilin on top of it, we have a full recovery of channel activity in less than a minute, or in about a minute, which is quite impressive. And on the next graph is we did the same experiment with TTX, which is tetrodotoxin, and here we don't have any recovery from saxophilin, which shows that this saxophilin rescue and this saxophilin effect is very specific to the toxin saxitoxin. And so this is the proof of concept, as Dan mentioned, that a protein can play this role of sequestration as a sponge protein to protect the channels against a toxin. And on the next one, I will pass it to Dan to summarize the story.
All right, so this goes back to this question of how do you resist a toxic environment? So as Jack pointed out, there's really three strategies that one can imagine. And the first is that you destroy the toxin, which is useful, but not useful if you want to redeploy the toxin as a way to protect yourself. The second is that you can make small changes that change the shape of the target ion channels so that they don't interact with the toxin. And that works, but that also comes with the cost. So every time a protein changes its sequence and acquires mutations, it may change the function, but it actually may change the original function. And that's what we found here, is that mutations that had been proposed to affect batrachotoxin binding actually were really detrimental to protein function. And so you need to think about the third way, and the third way is to suck it up, Kermit and Tweety, and that is to develop some kind of sponge that lets you suck up the protein, or suck up the toxin, and maybe even transport it somewhere else. Because you have to remember that those poison frogs and those birds, they get these toxins from the food that they eat in the wild. And so they need to move that toxin from the gut to their skin or to their feathers. And so there has to be a system to do that. And we think that that may be the role that these toxin sponges like saxophone and perhaps something called batrachophilin, which is a thing that we imagine must exist in these birds and frogs, but that we haven't found yet, but we'd like to look for. And we think that the study that we've shown really points researchers in a different directions to start to think about what kind of systems of sort of toxin sponges might be out there to protect animals from toxic agents that they encounter in their environment. And I think this is really general, not just for these really neat creatures that use poisons to defend themselves, but also for other animals that do encounter toxins in their environment and need to figure out how to deal with it. So we go to the last slide, I guess. We need to thank our great collaborators. I want to thank especially you, the American taxpayer, because you guys foot the bill for everything we do in the lab. The support in my lab was supported by the National Institutes of Health and the Department of Defense. We've had fantastic collaborators at Stanford, Justin Dubois' lab and Lauren O'Connell's lab, who bring expertise in toxins and poison animals, as well as Megan, who started this project with Jack and brought it to us with this great question of how do the bird sodium channels carry mutations? We started this by thinking that that would be the answer. And I think the beautiful thing about science is that sometimes you hit a dead end and you have to figure out a way around it. And that this was really a perfect example of the scientific method in action, because the first hypothesis we had, which was that these poison animals would have batrachotoxin resistant sodium channels, turned out to be wrong. And so we needed to figure out how the animals still survive. So
yeah, so I don't know if Daniel remember my disappointment when I found out that these channels were sensitive to the toxin, and our disappointment after that. But it didn't last for too long till we figure out that there was something more interesting behind that data, actually.
Yeah,
and I'd say too that there was a lot of work that Fayal did in the lab to actually really nail that down and to be absolutely positive that the channels, were functioning and that they were poisoned by the toxins. So there's a really cool set of experiments that Dan and Fayal were able to do in the lab.
Yeah.
So
yeah, this was very nice.
I think the best thing here is with Jack and our other collaborators who all bring different kinds of expertise to the problem. It's really a good example of team science, and actually why sort of this curiosity-driven science can be a lot of fun, and what makes it great to be a scientist.
Mm-hmm.
So thanks, and I think there might be some time for questions if we want to turn it back to Lynn.
Alrighty, popping up here with Jack and Fayal. Thanks for such a great, robust presentation about this amazing work that goes so deep. It's so amazing. First, we've got a comment from Julie about that last slide with the bullfrog and sucking it up, saying, I like this slide. I also like that slide. And could you actually tell us a little bit more about how the sucking it up mechanism might have medical implications moving forward, just in general?
Yeah, well, one of the things that we've learned with the birds is that they have some mechanism for moving the toxin into the parts of the body that they want it to be and not where they don't want it to be. And so, there's these ideas of chaperone proteins or, some sort of a binding protein, and it can have multiple uses. And one of the things that's cool is we know so little about these kinds of proteins. And if we can find in the birds and in the frogs what these proteins are, then we can learn a little bit about how they bind other proteins and move them. And we could even potentially use those proteins to, if somebody gets poisoned by these, we would have a ready antidote. And poison dart frogs are able to resist a huge variety of alkaloid poisons. And so potentially, either there's one or many of these different proteins like saxophone we'd have a whole arsenal of chemicals that you can use to soak up different alkaloids.
Yeah, that's really amazing. And another question, since we've got so many rolling in, do pitohui feathers / skin retain some of this toxicity even after death, like in museum collections? And that kind of goes into another question about how you tested bird tissues. Like, could you tell us a little bit more about how all of that works together?
Yeah, it turns out that botryga toxin is a pretty robust chemical, and it doesn't break down very readily. And in fact, my colleague John Daly, who studied these chemicals long before I did, and who helped isolate and identify them from the birds, had gone back to 100-year-old dart tips and found that they were still poisonous in the Smithsonian's archive. So that's pretty amazing if you think about it. And we'll often like pluck a single feather from a bird, and then you can put it on your tongue and taste, to sort of see how poisonous a bird is. And you can bring those back into the laboratory in a Ziploc baggie, and and they'll be poisonous for a long time, enough to taste that.
Wow. Yeah, that's amazing. And yeah, thank you, Kat and Julie, for those questions. And one last one for Fayal, actually, directly. Since you were so hands-on in the lab with all these frogs and, working with all of these organisms as part of this work, like what did a day in the lab look like for you? Can you tell us about it?
Yeah, I did. The lab is quite exciting because especially when I was working on this project, when we started thinking about these new ways of for the frogs to protect themselves, then the lab would be when we receive this little beautiful poison frog, which are like 2 or 3 inches long. They're tiny, but they're colorful and beautiful. We'll be receiving them, and all the excitement in the lab when we receive them. But then when I start doing the experiment, everybody in the lab will come and check on the frogs, what's happening, because in this challenge experiment, the idea is to see whether the frog are going to resist the toxin or not. And so the xenopus, which is a much bigger frog, will not resist the toxin, and then people would be very surprised to see like these tiny little frogs will just be fine with huge amount of toxin, like 20 times the dose that should kill them, that should kill animals of their size. So that's an example of excitement in the lab, and yeah, it's pretty cool.
Yeah, I mean, I would also get super invested in frogs like that, especially seeing such a big difference in how the big and little ones react. Yeah, that's really, really cool. But I'm keeping this pretty short because we've got such an amazing program also coming up. But thank you both so much for your time. This has been amazing, and I'll pass it back to Lynn to pass back to our next guest, which will be Shannon Bennett.
Thank you.
Thanks for having us.
Yes.
Hi, folks. I'm really happy to be here and speak to you all about my work. I am looking at all the places that people come from, and I'm so impressed by how broad you're spread out around the world. I'm going to be talking a little bit about the origins of viruses and infectious diseases from nature in general, but all of us have been affected, hopefully not infected, affected by COVID-19, which is something I will include as I talk about this topic, because we're all poised to understand the origins of coronavirus, COVID-19, and what we could have done about it and what we can do about it in the future. So I want to talk a little bit about the natural origins of emerging infectious diseases, and particularly how, as we interact with biodiversity, with natural systems, how we could increase, or even by protecting biodiversity, decrease our risk of emerging infectious diseases. And I am going to tell you a little bit about my story. I have a personal connection to this topic. When I was a college intern, I went to West Africa and worked on some public health projects. And I'm just like all of us in this virtual room, I became a host. And I became a host to a malaria parasite called Plasmodium falciparum. on the left, Entamoeba histolytica in the middle, and a Staphylococcus aureus infection. And all of these took down my host defenses, spilled over into my system through natural interactions that I had. And they're all zoonotic or non-human animal in origin. So for me, that really hit home the importance of studying the role of zoonotic or natural origins in emerging infectious diseases. There's a lot of viruses and other diseases that I mentioned out there that have zoonotic origins. In fact, most viral diseases of humans have non-human animal origins, and I've named a few famous ones here: HIV, influenza, hantavirus, Zika, and many bat viruses. And we've all probably heard about Ebola and Marburg, but probably the most significant one that comes top of mind for us today is COVID-19, or the agent of COVID-19, which at the time was just classified as a novel coronavirus with the date 2019. And we now know that to be SARS-CoV-2. So I'll talk a little bit in a minute about where that came from in terms of its zoonotic origins. But first, I just want to talk a little bit about this idea of zoonotic spillover. So there are many sources, many ways we interact with non-human animals that can be a potential source of a spillover event. And there are many kinds of spillovers, exchanges that we might have with zoonotic reservoirs. And so on the left are these circles that show sort of the relative proportions of sources of spillover events from non-human animals. Most of the viral spillovers that cause diseases in humans come from domestic animals. And domestic animals could be chickens, ducks, as in the case of avian influenza. They could be mosquitoes in the case of West Nile virus or Zika virus. They could be mosquitoes that become domesticated. It could be pigs in the H1N1 swine flu pandemic. There are other kinds of sources like peri-domestic wildlife, wildlife like bats that might come and feed on fruit orchards around pig farms, for example, and that's a major pathway for Nipah virus. And there also are many captive or managed wildlife or farmed wildlife that could be a source of spillovers, and many of those are considered probable bridge hosts for SARS-CoV-2, which I'll get to in a minute. And of course, there are free-ranging wildlife like bats that can also be a source if humans come into contact with wild bats. And so then on the right are kind of boxes that talk about some of the kinds of spillover events. There are ongoing spillover events where where animals provide, non-human animals provide ongoing sources of exchange of viruses with humans, and that's in the red box. Then we have new things that crop up, which we define as emerging infectious diseases. And this is where SARS-1 and SARS-2 both fall, as well as things like Ebola virus or West Nile virus and Zika virus. And then sometimes we actually infect non-human animals back, and that's a zooanthropognosis. And actually that's happening with SARS-CoV-2 too. It turns out that if you heard about the minks in Denmark that were infected by humans, that's an example of a zooanthropognosis. So the take-home message here is that we are interacting either directly with nature in terms of free-ranging wildlife or with hosts that come between us and nature, but they themselves definitely interact with nature. And all of these pathways could arise in viral spillover and cause a range of different kinds of diseases. My focus is on the emerging infectious diseases. Where are the sources of new things? So it turns out that there are abundant viruses in our world. And this is a characterization of the virosphere. And what I'd love you all to take away from this is an impression of all the different colors and shapes and sizes and ways of making their way in the world that is the virosphere. So it's an incredibly diverse and up to this point barely understood source of incredible diversity. There are only about 6,000 viruses described out of probably up to 1 trillion potential forms out there in nature. So my work at the Academy, which again started when I was an intern in Africa, thinking about zoonoses and how I was interacting with the natural world to become a host, I really wanted to use mosquitoes to give me and my lab insight into the diversity of the virosphere, focusing in on the virosphere that mosquitoes might interact with. And so I have my daughter sampling mosquito larvae on the left, and I'm in the middle sampling mosquitoes with nets. We then take the mosquito, which you see in the bottom panel, filled with blood like a little red jewel, and we pop it into a tube, and then we bring it into the lab, and we process it in a way that cracks open the genetic information in that drop of blood, as well as in the mosquito itself. And we can find viruses that we already know about that are mosquito-borne viruses like dengue and Zika and West Nile, but we can also look for new viruses. We have the genomic technology at the Academy to look for the genetic signature of viruses that we don't even know about yet. We have a lot of precedence in mosquito-borne viruses. We know that many of them evolve from non-human systems. And this is an example of work we've done in dengue. I mentioned Zika. Chikungunya is another mosquito-borne virus. And all of them have non-human primate and wild mosquito origins, and those are shown in pink. And then the non-colored tips are the cases in humans. And so we know that mosquitoes are really important sources of viruses, and importantly, they can bridge a natural system like a non-human primate community and a human community, especially as those mosquitoes become domesticated. So as we continued to explore these mosquitoes, we found new viruses. The new viruses we found are all in yellow, and they're related to viruses that cause disease in humans, and those are represented by the skull and crossbones of these family trees. The slide before and this are showing, based on genetic information, the family trees of viruses, just like you would develop a family tree of your brothers and sisters and cousins related to your parents, your grandparents, and your aunts and uncles. So they're family trees, but instead of using your own family history documentation, we're using genetic information to reveal relatedness. So we can see that many new viruses, which we're still just beginning to describe, are genetically related, connected to viruses that cause diseases in humans, as well as other animals. So that really takes us back to COVID. I'm setting up a precedent. We know that there are rich natural reservoirs of viruses. And we also know that there are multiple pathways that have occurred connecting natural viruses to humans. And so using some of the mosquito-borne viruses I've worked on, but as well as thinking about the origins of COVID, our job really is to track those pathways down so that we can understand how our relationship with nature is somehow forming a bridge to facilitate these emerging infectious disease events. So I want to zoom into the COVID-19 outbreak. So just in case you don't know, COVID-19 refers to coronavirus infectious disease. That's the COVID part. And then the 19 refers to 2019. A lot of people joke that it happens to refer to the number of pounds we all put on when we had to lock down and stay home during the COVID outbreak, but it does not mean 19 pounds. It actually is related to the date. So COVID-19 at the time when it was first noticed in December in China, December of 2019, was not related. It wasn't named as a virus. It was named as a syndrome. So COVID-19 is the name of the disease syndrome before it was really identified as with an agent, a viral agent, a specific viral agent. The other thing to note in this chart, which is basically a map of Wuhan, China, which is where the first case clusters were identified, is that they were clustered around a wet market called the Hunan Seafood Market. And many of the cases were associated with this market, but later on it was discovered that there were other wet markets that were also involved. And then the visual with the little blue boxes are actually the tents of that seafood market just before it closed down. So those early cases gave rise to actual samples that could be sequenced. And using that genetic information, those viruses were able to be placed in a family tree of relatedness. And that family tree drew the closest connections, the strongest relationships of SARS-CoV-2 to a set of bat viruses, which were named the bat precursors. It turned out that this virus is called SARS-CoV-2 because it was also in a branch of the family tree that put it closely related to SARS-1, which was a major outbreak in 2002, 2003 in humans that shut down, had a world impact, shut down airports all over the world. I don't know if folks remember the immediate effect of SARS-1 emergence. So that helped us identify this COVID-19 agent as SARS-CoV-2. There have been 9 viruses that have emerged into humans, SARS-CoV-2 being the 9th, over the last 100 or so years, and 7 alone in the last 20 years. And these include MERS, Middle Eastern respiratory virus, shown in purple near the bottom, as well as at least 4 distinct lineages of human coronavirus, which these are viruses that cause anywhere from 10 to 30% of all common colds. So they jumped over into humans and also cause disease. And in some cases, we haven't identified their bridge hosts either. But I think basically what this family tree shows you is that the ultimate origins of COVID-19 is a SARS-like coronavirus called SARS coronavirus 2, and the deep time origins are clearly placed in bats. So I mentioned Ebola and Marburg, Nipah, Hendra. These are all viruses that actually have ultimate origins in bats. This is one of the bat hosts that hosts relatives to SARS coronavirus 2, and it's called the intermediate horseshoe bat. It's pretty cute. I love bats. So, but what I really want to talk about is that question I posed about really actually tracing the pathway between nature and humans for this coronavirus. And so I'm calling this sort of the proximate origins of SARS-CoV-2. And some of the early investigations that I want to share with you were actually recently published in a paper by Eddie Holmes and colleagues, and it just came out earlier in August. And it shows 2 things that are really important in tracing that pathway back to nature. One is that it shows that the earliest samples, and that's in section A, the earliest samples of coronavirus in humans, SARS-CoV-2, were actually pretty diverse. There were at least 2 distinct lineages, an A and a B, and they were co-circulating as early as early January 1. And actually even into, as early as January 1. And so that suggests that there were actually probably multiple interactions between humans and an animal reservoir that caused multiple spillover events. The other side of this figure in panel B shows the cases that have clustered in space, not and time, and they all cluster very close to this Hunan market, as well as several other wet markets. Now, also shown on this chart is the Wuhan Institute of Virology, and it's quite a bit south of the Yangtze River, and it's designated WIV. And the reason that this is included in this figure is because there's a lot of speculation that it's possible that this virus could have emerged from a lab accident. And I'll just tell you that the scientific community, based on the data presented here on the next slide, are pretty much unanimously against any plausibility behind that hypothesis. So another piece of information that's really important in tracing the pathway between natural systems and humans is this figure, which dives deeper into the closest relatives to SARS-CoV-2. And so there's a family tree again in the lower right-hand corner, and you can see the little red spot beside the SARS-CoV-2 label derived from humans in 2019. But clustered very closely to this virus are a bunch of other bat viruses. And those bat viruses are really distributed, those samples were taken quite broadly. So you can trace those colored spots to the map on the sort of upper left. And you can see that there's the SARS-CoV-2 emergence close to Wuhan in red, but we also have a bunch of bat viruses that are scattered throughout the entire southeast and south parts of China. And so Yunnan has often been called out as a potential origin for this virus, but it turns out that there are relatives at rat G13, which is the virus that was also described that was described by the Wuhan Institute of Virology, is actually not the closest relative, and there are many close relatives scattered throughout southern and southeast China. And so it suggests that somehow this virus, which is widely distributed in nature, emerged multiple times into humans and had to travel a distance of about 1,500 kilometers to get there. And so that really points to a missing link. Basically, most probably similar to SARS-1, some kind of an intermediate and non-human animal host that picked up this virus through exposure to bats and then transmitted it to humans. And if you remember the story of SARS-CoV-1's origins, that was a civet cat. It turns out that civet cats, raccoon dogs, and a range of common mammals that are in these wet markets, sold actively in these wet markets, and even farmed for their fur on a very large scale, are potential easily infected by the SARS family, and very likely hosts, bridge hosts between bats and humans for the SARS-CoV-2 emergence. So you may all be thinking, wow, how often do spillovers really happen? And it turns out that they happen a lot more often than we realize. So this is a service that reports, that scrubs many news sources as well as public health reports in many countries all over the world, and posts alerts. And these alerts are often reflecting a spillover event. So just today I got an announcement that there was a Marburg spillover event in West Africa, in Guinea, that is actually new to that region. We're not used to seeing Marburg spillover into humans in that region. And so spillovers do happen all the time. And so they're definitely, we definitely need to pursue that missing link between human and natural systems to understand better how to mitigate these spillovers that do happen, but they don't always translate into an epidemic or a pandemic. So I want to bring you back to this graph just because I want to talk about what the product of my research has led us to conclude. And that is that there are many different animal sources, non-human animal sources for zoonotics, but what ties them together? And I had mentioned that domestic animals, including domesticated mosquitoes, could play a huge role. They do play a huge role in some of the known zoonoses and emerging infectious diseases that we know about. But importantly, they definitely played an important role in COVID, SARS-CoV-2 emergence, because we've clearly, although we've linked it to bats and free-ranging wildlife, the distance traveled and the multiple events and the link to the wet markets suggests that there's a huge role for either captured, managed, farmed wildlife to have driven that event, or even domesticated farmed animals like mink or red fox. So this brings me to some of the work that we want to do going forward, and that is to really develop a big high-level synergistic view of these processes and what we can do to identify them before they become an emergent event. And so I talked a little bit about the role of agriculture, mass farming, domesticated animal farming, and even urbanization in the case of domesticated mosquitoes, in providing a bridge from the natural diversity of the virosphere into humans. And that's what I want to talk about with this figure. And so what we're really trying to do is come up with a signature and a footprint of what the hallmarks might be of when a virus that's in natural systems could emerge to become an epidemic or a pandemic. And we're pretty sure that the link to agriculture or to urbanization is truly one that is not only extractive to nature, but also helps to drive emerging infectious diseases from natural sources into humans. So what can we do to sort of prevent that, or at least get ahead of it? So a lot of our work in the mosquitoes, as I told you, is revealing the hidden virosphere in these animal systems. And so on the far left, I have the hosts like monkeys and guinea fowl and mosquitoes shown in the gray bars. And we're now appreciating that they contain a wide variety of viruses. And those are shown with the colored bars. What happens as you shift your landscape to be heavily agricultural, where you have monocultures of certain kinds of domesticated animals, like mosquitoes in an urban environment, certain mosquitoes like Aedes aegypti, rats in the case of hantavirus emergence, or even corvids and the common house mosquito in the case of West Nile virus, is that you actually skew not only the hosts, but the viruses. And you get a virus like in this high red bar in the middle that basically causes an outbreak. It becomes ecologically and evolutionarily specified to this new host monoculture or host niche. And viruses we know can evolve very quickly, and that can cause a pandemic. And so what we hope to do, we recently received a major grant from the National Institutes of Health to try to scan these natural systems as well as peri-domestic and domesticated animal systems to understand where viruses might occur along that landscape gradient and detect novel pathogens early before they've really built up that number to become dominant in a human system. And so we've focused on 3 countries, Nicaragua, Ecuador, and Sri Lanka, but we're part of a global network that focuses on countries all around the world, in South America and Southeast Asia and in Africa. And we're using genetic signatures to discover novel pathogens and describe their population dynamics as well as their evolutionary changes, just like we're doing with Delta virus or other COVID virus variants, where we're looking for new signatures of an adaptive virus that can either escape the immune system or replicate really fast or transmit really fast in humans. So stay tuned for that. I hope to give you more as the data unfolds. This is the network that alerted us to a Marburg outbreak just today. And so we're staying tuned as a network all over the world to do early detection of outbreak events, as well as look for those hallmark signatures of adaptation and emergence. And with that, I would love to entertain some questions.
Hi, Shannon.
Hello.
We have a few questions. Let me pull down your slides. So the first one is kind of like a 2-3-parter.
Sure.
Why mosquitoes? Firstly, like why did you gravitate towards studying them? But then also why are they such a prominent carrier? And then Tom also is asking, what is a domestic mosquito?
Right. So why mosquitoes? So if you recall my Africa origin story, where I was a host, an unwilling host to a lot of pathogens, the main one in that spectrum and the first one to hit me and the one that has major impacts around the world is Plasmodium falciparum, or malaria, and it's transmitted by mosquitoes. And so that definitely got me thinking about mosquitoes. There are over 6,500 mosquitoes around the world. And most of the mosquitoes that can carry malaria are wild mosquitoes, and they don't necessarily live around human communities. But some have become super adapted to living around humans. Some malaria mosquitoes, as well as the major vectors of dengue and Zika, which is called Aedes aegypti, or the yellow fever mosquito. And that's become very well adapted to humans, so much so that it can't live anywhere else except around human dwellings. It's a very weak flyer. It can really only move from your bedside to your closet to your potted plant water tray, and that's about all it's good for. And so I consider those kinds of mosquitoes domesticated mosquitoes. It's not that we domesticated them on purpose, but we did domesticate them by accident. Great question.
Yeah. You were kind of speaking of the different transmission roles. There's a question about, like, are there any instances of plants playing a role in virus transmission, either with humans or non-humans?
That's a really great question. Actually, it turns out that one of the major hypotheses out there about the deep time origins of viruses that are mosquito-borne might be that they actually started out by being as plant viruses that were picked up and transmitted by plant tissue sucking insects like aphids. And so plants were some of the most abundant early eukaryotic hosts on our planet. So it makes sense that they have a rich viral diversity. Indeed, they do have a rich viral diversity. And some plant viruses definitely land in the family tree of vertebrate and mosquito-borne viruses. So another to be continued, stay tuned kind of story is emerging as we discover more and more of the virus sphere and where it exists in nature.
Great. And then last question. So based on what you showed with the forest to urban environments hosting different species, it sounds like biodiversity is a really important natural barrier to preventing huge virus transmission. Could you just tell us a little bit more about that?
Yes. So there's a hypothesis out there called the dilution effect. It's just one hypothesis, and it's not a particularly evolutionarily oriented hypothesis. It's more thinking about, it was developed, strongly advanced in thinking about the origins of Lyme disease and where it might impact humans the most. And Lyme disease is caused by a bacterium that's a host generalist, and actually it can move between different tick hosts as well as small mammal hosts and even big vertebrate hosts in its life cycle. And so originally the dilution effect was considered important. If you had diverse hosts, it wouldn't pose an undue burden ecologically on any one host like a human, and it also wouldn't pose an unduly attractive evolutionary target because there wasn't like one single host. It's kind of like when people develop mixed small crop farming and agriculture, and the idea there is to avoid a big bullseye on your corn or your big monoculture of industrial agriculture for plant pests and plant pathogens to emerge. And so there is this sort of evolutionary opportunity that viruses can rapidly take advantage of when you have an overly dominated, skewed community of hosts. And that is one of the hallmarks of biodiversity loss, is you get communities that are basically characterized by only a few highly dominant hosts. And so not only do they become an ecological target for a lot of potential pathogens, but also an evolutionary opportunity for fast evolving pathogens.
Yeah, that makes a lot of sense, kind of the way you've framed it. Well, thank you so much, Shannon. It's always such a pleasure having you on Night School. Yeah, thanks again, and we look forward to hearing more about your research.
Yes, thanks for having me, and have all a good night. Be safe.
Thanks.
Bye.
Up next, we have Jessica Hernandez.
So good evening, buen día, Padiche Buguri. My name is Dr. Jessica Hernandez, and today I will be presenting on Indigenous science and climate change. So before I begin my presentation, I would like to virtually acknowledge the lands where I'm presenting from. Today I'm virtually presenting from the ancestral lands that were and continue to be the home of the Kappa and Osage people, now known as Little Rock, Arkansas. I also want to acknowledge the ancestral Black history that resulted in the civil rights movement that was carried out as a result of segregation that existed in the city and across the country. A little bit more about myself. I'm a transnational Indigenous scholar and scientist and community advocate. My STEM background ranges from the marine sciences to forestry, and through Western science and Indigenous science, I'm able to advocate for food, climate, environmental, and energy justice. And my book, Fresh Banana Leaves, Healing Indigenous Landscapes Through Indigenous Science, is forthcoming this January 2022. So what is Indigenous science? Indigenous science is our indigenous ways of knowing. It's knowledge that has been formed and formulated since time immemorial. It's essential knowledge that we are able to carry to the future generations. For me, Indigenous science is also a holistic approach that allows us to connect humans from nature. And this is something that we are taught since we're kids. And with these conversations also comes respect, responsibility, and reciprocal relationships to nature. So what that means is that for us, animals and plants tend to be our relatives. So we have those respect and responsibilities that we have to carry forthcoming when it comes to nature. And Indigenous science is a reason why, despite only making 5% of the world's population as Indigenous people, we are able to steward 80% of the world's biodiversity. And I think it's important for me to mention that what is now known as Latin America, as a transnational Indigenous woman who comes from Latin America as well, that 50% of the world's biodiversity is currently located in Latin America. So what this means is that Indigenous peoples from south of the border get to steward most of that biodiversity of the world. For me, Indigenous science is also a medicine because it begins with the understanding that our relationship with our environment, it also determines the cosmos, determines our well-being. So if you have heard of Indigenous cosmovision or Indigenous traditional ecological knowledge, that's the same as Indigenous science, but I rather prefer to use the term Indigenous science. And I really like this infographic because with Indigenous science, we learned that humans are not separated from nature. And as we can see, when we often see life cycles for many animals, we don't see humans embedded in those life cycles. And I think that our way of knowing our Indigenous science allows us to understand that within those life cycles, with the water cycles, everything that has a spirit, that has a living species is also connected to humans. And I think that that's one of the big differences between Indigenous science and Western science, is that despite us having conversations that humans are connected with nature, when we talk about sciences, when we talk about, for instance, salmon and draw the life cycle of salmon, we tend to forget to incorporate humans. And that is an important thing to mention in how that differs Indigenous science from Western science. As I mentioned, another word that for Indigenous science is also traditional ecological knowledge. And Dr. Wildcat, who is the author of the Red Alert, mentions that, TK, traditional ecological knowledge holders, we acquire our knowledge intergenerationally. And that's through lifetimes of attentiveness and mindfulness to the places that we live. And as a result, Indigenous science is place-based. So for instance, as an Indigenous woman, I have that Indigenous science to the ancestral lands where I come from. And I think that that's important to also mention when discussing Indigenous science. So Indigenous science kind of is integrated within our history. So if you know about the Mayan ruins, it's important to mention that despite them being called ruins, they continue to be sacred sites and also traditional sites for the Indigenous Maya people that currently live today. Also, we see how our Indigenous science is also replicated in our regalia, which is our traditional clothing that we wear for ceremony or traditions. And oftentimes our regalia reflects our nature, it reflects our place-based environments. And I think that's also important. And obviously with our Indigenous science, we're able to understand how to use our food systems, our native species, those that food sources that are locally grown and are available for us in our communities and environments. And when I think about the biggest contributions that indigenous science has had to modern times, I think about the arts, math. Obviously, we know that the number zero and all these mathematically laws came from the Mayan civilization. We know that indigenous science resulted in rubber, and then astronomy. Our knowledge of the astronomy that currently exists today is a complement of indigenous science that has been passed down through the generations. We also know that the writing system is as a result of that indigenous science that is still present in our communities. And obviously another one that people love is chocolate, because chocolate did come from indigenous communities, like the being able to use cacao, which is a native species, and make it into chocolate. It's an indigenous practice, and that's the way that indigenous science kind of formulates itself into modern days, despite it not being recognized as that. And despite all of these contributions and other contributions, because obviously there are more, our indigenous contributions are ignoring Western science. And I think that when we talk about climate change, we need to start questioning whose science is validated and whose science is often ignored. And, one of the questions that I always ask everyone is like, who are the scientists that are, invited to the table, who we continue to learn from, who are taught in those introductory courses that we take as scientists emerging into the field. And obviously there is no indigenous voices or perspectives or that indigenous science that has been sustained throughout the Americas as we are on indigenous lands, no matter where we stand from. And as I mentioned, it's important to emphasize that our indigenous science is also our indigenous spirituality. So like I mentioned, even as children, we learn that our animals and plants are our relatives, that everything is holistic, that everything is a part of who we are, and we are not a part of nature. Like we're an entire ecosystem. And this is how we're able to sustain our cultures, our identity, which is our indigeneity, because we continue to steward that environment and continue to be immersed into that environment. And we all know that the IPCC report recently came out, and according to the Department of Economic and Social Affairs, climate change continues to pose the biggest threat to indigenous peoples, even though we contribute the less to the greenhouse gas emissions as compared to other communities. And I think this has to be highlights the inequities that exist when we talk about climate change and indigenous peoples. The first inequity is the degree of responsibility for climate change. And I think that quote kind of relates to that despite us not being as responsible for the CO2 emissions, for the fossil fuel, for the extractive resources that continue to alter the climate change, the CO2 rises, we have that bigger responsibility. And as I mentioned, that responsibility comes because that's the way that we're taught, right? It's the indigenous science that we have the responsibility to protect nature so that nature can protect us. And despite that, we have the largest responsibility because climate change continues to impact us. Inequity, too, is that climate change impacts are higher in the global south. And as I mentioned, in Latin America itself, like south of the border, 50% of the world's biodiversity is currently located there. And the inequity 3 is that the ability and capacity to deal with the impacts of climate change. When we talk about the funds, the grants, and every other resources available to combat climate change, indigenous communities don't have those resources. We're often the last ones to be able to tackle those resources or getting of those resources, but yet, we face climate change first and the highest. And the inequity 4 is the intergenerational impacts of climate change. And that's something that I go more in depth in my book, and I'm going to be mentioning in this presentation as well. And inequity 5 is the gender disparity of climate change. And it's important to mention that climate change impacts indigenous women the most because we do carry that responsibility in our roles in our indigenous communities, our pueblos, where we have to respect and teach our children. We nurture that nature that is passed down through our generations as well. And I wanted to mention that it's important to note that indigenous peoples are not monolithic. So there's differences based on our intersectionalities. And like I mentioned, indigenous science is place-based. So it's important to mention the location where we come from. And climate change impacts each indigenous community's tribe and nation and pueblo differently. So I'm going to be more focusing on the ways that climate change impacts my local communities, my personal communities that I have those kinships with. And indigenous peoples and the role that they play in addressing and combating climate change are rarely considered when we talk about climate change in science. And I hope that this presentation starts allowing us to question why that tends to happen, why despite there being indigenous science, that knowledge that we hold that has been generated and formulated since time immemorial continues to be ignored within the climate change discourse. And like I mentioned, that we continue to be rarely considered in public discourses of climate change, climate policy, and everything that has to do with climate science and the continuation that we continue to face. And it's important for us to start uplifting indigenous peoples and voices and bring to the forefront that science. So despite that, indigenous peoples, we're vital and active in the many ecosystems that inhabit our lands. And this is why we're able to steward 80% of the world's biodiversity despite only making 5% of the world's population. So just noting that we are 5% of the world's population, yet we steward 80% of the biodiversity, that just shows the strength that indigenous science, indigenous ways of knowing carries. And it emphasizes that it is important, especially when we talk about climate change, because we want to protect our world's biodiversity so that we can have that and sustain that for the future generations. And I think that oftentimes it's hard to think of the future, but as indigenous peoples, that responsibility comes from us thinking about the 7 generations that come forward from us. And every day we're taught to walk as a future ancestor, right? Because we all end up leaving this earth. And I think that it's important for us to keep in mind that it might not impact us as much today, but it's impacting indigenous peoples, and obviously it's going to impact the future generations. So I wanted to kind of focus more on my community. So as a Maya Chorti woman, indigenous Maya people, I'm going to focus on what we face. So we continue to face forced displacement. And I think that's important to kind of go back into not history, because this is like through my parents' and grandparents' generation. We faced a war that was known as the Central American Civil War. However, this war ended up being a genocide. In El Salvador, this war took place from the 1980s to the 1990s. And in Guatemala, it took place from 1960s and ended in the 1990s. And it's important to note that even though there's these timelines, it began before those years, and it kept on going for those years. And why I say that it was a genocide is because it has been declared as a genocide, because during the Central American Civil Wars, indigenous children were the ones that were targeted. And I mentioned El Salvador and Guatemala because my Maya Chorti community is separated by those borders. So these are pictures, like historical pictures that account how our indigenous children were forced as child soldiers in both Guatemala and Salvador. And I think that that's important to mention, because as we continue to face climate change and all that settler colonialism, right, that impacts our indigenous communities, our indigenous children are the first impacted when it comes to those phenomena. As a Maya Chorti woman, oftentimes I hear that the Mayan civilization is extinct, right? Because oftentimes we talk about the Mayan people as like people of the past, people that no longer exist. And in archaeology, we have this classic narrative that the Mayan civilization collapsed, so therefore there must be no Mayan people today. However, indigenous Maya people continue to exist. However, we continue to face that erasure. We continue to be dismissed and ignored even in our own ancestral lands. For instance, in Guatemala, the indigenous rights was adopted into the Constitution in 2010. In El Salvador, the Constitution amended to recognize indigenous peoples in 2014. And I mentioned these 2 countries because, like I mentioned, my nation is separated by those borders, and also because oftentimes what we know, most Maya people live in those countries as well. And on top of that, we have also faced land grabs. So land grabs are basically when the government took most of our lands and sold them. And they ended up selling them to agricultural corporations, plantations. So that's why when we talk about bananas, when we talk about coffee, most of those resources come from Central America. And that continues to impact our indigenous peoples because we are used as cheap labor, right? We continue to sustain these capitalistic entities that are destroying our lands because they're extracting, for agricultural purposes so that they can import most of these resources, to the point that even our people who sustain these resources are not able to afford them. And I think that this is important to mention because this also contributes to climate change, and it also contributes to our forced displacement from our native lands. And something that I refer to as the climate change intersection with settler colonialism is eco-colonialism. And what I define eco-colonialism, and it's something that I mentioned in my book, is that non-Indigenous peoples govern our natural resources and Indigenous lands without consulting us Indigenous peoples, and that respect Indigenous sovereignty. The severe altering of our landscapes due to settler colonialism and the ideologies that were introduced as a result of that. For instance, climate change impacts and the lack of resources offered to Indigenous communities who are already experiencing the impacts of climate change. So eco-colonialism kind of relates back to those inequities that I mentioned, how climate change continues to impact Indigenous peoples. And another thing that we face as Indigenous peoples south of the border is that our Indigenous leaders continue to be murdered. We face the highest violence. So as a result, for instance, in 2019, 28 Indigenous rights defenders, land protectors were murdered in Latin America. In Mexico in 2020, 28 environmental leaders were murdered. And then in the Amazon, in the Amazonia, one Indigenous rights leader is killed every 2 days. So that shows the violence that we continue to face even when we uplift our voices to protect our lands. And I was once asked, what is the hardest thing about my field of study? And that's knowing that for the work that I do, being safe in the United States, my people back in my ancestral lands are targeted for that. And I think that that's something that I hold deeply in my heart. And that's why I continue to uplift Indigenous voices, Indigenous science when I talk about climate change and when I talk about my work and the research that I continue to do. And as a result of all of those things that I mentioned, this is why we have that forced displacement. If you have heard of the immigration caravan, a lot of those people are Maya people. They're being displaced from our lands because of those land grabs, the plantations, the exploitation of our lands, the violence that our leaders continue to face when they try to speak up to protect our lands. And this is why we're seeing this wave of refugees, and they're climate refugees because on top of that, the climate change impacts that we're facing. For instance, last year we faced both hurricanes, and the Indigenous Maya communities were severely affected to the point that when the government provided resources to address those impacts, we were the last ones to receive any, if anything at all. And I think that it's important to mention how climate change is resulting in this forced displacement. And oftentimes when we talk about immigration, it's kind of criminalized. And when we talk about our identity, we're often labeled as Latinx or Hispanic, when in reality our Indigenous communities are not a part of that Latin narrative, because actually we're still oppressed in those settler governments that Latin America operates under. And Indigenous science is our cosmovision, the way that we see the world, our spirituality, our medicine. And it's important to mention that they act at the same time, the myth and the history. It's a part of our death and rebirth as people and as spirits that continue to live on these lands. And this, climate change for my communities is impacting our displacement. It's also impacting our language, because oftentimes when we are displaced from our communities, the younger generation doesn't get to learn our native language. And for us in the diaspora, many of our people have to reassert their indigeneity, the forced migration. And obviously, when we are in the diaspora, we have to reclaim our environmental kinships as we continue to thrive. So I think it's important to mention that, Indigenous peoples and the role that we play in climate change is rarely considered in public discourses in climate change. And I think that if we start reflecting on why that's the case, we can start to bridge those gaps and start to uplift that Indigenous science and those Indigenous voices that's important to address, especially when we talk about climate change and the impacts that it faces. For instance, COVID, right? We just had a presentation on COVID-19, and obviously COVID-19 impacted Indigenous communities the most because we already have those health disparities, and climate change and the pandemic is just exacerbating those health disparities. So disparities that already exist in our communities, but we were able to use the Indigenous science, and that's why our Native American reservations we came through when it came to the vaccination rates, why we were able to vaccinate our people at higher rates than other people. We were able to distribute those vaccines to even non-Indigenous peoples. And I think that's important to mention, because when it comes to Indigenous peoples, we work as a community and we use our Indigenous science that comes from our elders, from generations, to help one another, especially when times of pandemics and things like that. And this is just a quick overview, but my book, Fresh Banana Leaves, Healing Indigenous Landscapes Through Indigenous Science, goes more in depth of many case studies that continue to affect my people, especially Latin, Indigenous women from Latin America. And I hope that this presentation kind of brings awareness to why Indigenous science is important in those conversations that we need to start having. And that's all. Thank you.
Hi, Jessica.
Hey.
I was waiting to be popped on the screen there. Thank you so much for sharing. I was like trying not to cry in the corner over here, but yeah, just thank you so much for sharing and sharing your story, as well as giving us a little bit of insight into, like you said, obviously Indigenous people are not a monolith and not all the same, but just kind of these higher level things that are, obviously very common in how our society views and treats Indigenous people today. So yeah, thank you for that. And we have a bunch of questions, and the first one is actually, I'm really curious about how, so it's obviously extremely important to uplift Indigenous voices in science, like all around, and climate change especially. And I wonder, what are your thoughts on like how we can make sure to do that and make sure to acknowledge Indigenous science, but also not make Indigenous scientists subscribe to like the traditional formats of like scientific research and papers and all of that kind of publication formats, if that's not something that is well fitted to the science and knowledge that they're holding.
Yes, I think it's important to ask the Indigenous communities because, for instance, sometimes some certain knowledge, we want to amplify it to the point that we want to share it with non-Indigenous people. So a publication route will be the best approach. Oftentimes, like many of our people don't know how to speak or, even write. So even assisting them with writing their story, being able to tell their own stories, because instead of our stories being told and written for us, that's very important for us to navigate. And I think for my communities, one of the things is like even learning about the immigration discourses, right? Because oftentimes we talk about immigration, so we think it's a, brown issue, when in reality it's like more in depth, right? It's Indigenous peoples who are being displaced from Central America, especially Maya people. And when we talk about immigration, most of those resources are in Spanish. And most of our people don't speak Spanish, just speak their native language. So even seeing that and how that's an intersection that as scientists we tend to ignore because we're like, oh, that's immigration, that's more of the law, the policy side. But it's as a result of climate change and all those impacts that as Indigenous peoples we're facing back in our ancestral lands, where we reach the point where we can no longer fight because we face violence and harm. So we have to escape that violence that continues to exist even today.
Yeah, I think that's really well said. And so it's like a matter of having to, like, we need to protect the people, protect the land, and also as a result, this will like move us toward being able to bridge those gaps, right? Between like actually being able to like not siloing law from science because they're all very much interwoven. Yeah, so yeah, that totally makes sense. I really love that. And we actually have a question from Blake in the comments asking, is there anything that we can do to center Indigenous scientists and like specific actions we can take in our everyday lives and, things that we can do as scientists or non-scientists?
Yeah, one of the things that I have experienced in my personal trajectory as an Indigenous scientist is that oftentimes in Western science, we view Indigenous communities as an area of expertise instead of viewing them as stakeholders, viewing them as collaborators. So I think that even shifting that narrative where we're not trying to become experts on Indigenous science or Indigenous communities, but seeking those collaborators, seeking those stakeholders from those communities to become a part of our team, I think that's a way that we can do it. It's like, kind of unlearning everything that we have been taught and then relearning everything that we need to learn so that we can do it properly, and also follow those cultural protocols that exist, differently among various indigenous communities. I think that's a good way to start.
Yeah, like not making it like okay, so today we're gonna focus on indigenous science, but instead making it like a part of actually like just incorporating it. I love that. Yeah, I think that's really well said. Yeah. And let me see, looking through my list here. And I guess kind of at an everyday level, like not even from a science perspective, but I really love what you were saying about impacting generations ahead of us, and how that's kind of wisdom that we all really need to take with us, especially looking at the upcoming years with climate change, like here around the world, like all of the places. What do you have some advice on how, like, for those of us growing up here in this like very Western culture where it's like that, basically, what do you have any advice on like how to get to that unlearning process, and how to really like reshape the ways that we think of humans and nature as separate, like from your experiences?
Yeah, I think that starts with the K through 12 system, right? Because even when we learned about like life cycles, we tend to just draw, like for instance, if we draw in a fish life cycle, we tend to just focus on the fish and we never draw the human. So even questioning those little things that we were taught that were correct, and seeing how we can change it to actually incorporate people to begin with, right, in general. And then adding that layer to incorporate indigenous peoples will be a great way. And I think that also the younger generation that's coming through, right, the Generation Z, and how they're becoming social influencers because they're activists and they're actually caring about climate change. I think it's bringing those youth perspectives into the picture and learning from the youth, right? Because oftentimes, as adults, we tend to ignore the youth, but the youth also have a rich perspective that they can bring to the table, and they're also like already tackling climate change as we speak. So even bringing more youth perspectives when we talk about climate change, and not using like those credentials to validate or invalidate people's knowledge. I think it's an important way to start doing that, and I think the youth, especially with social media, have a great way to start. They are doing that already. So even listening to them and changing the way that we get our news, right? Because we can learn from social media as well.
Yeah, so. No, I totally, I love that, especially considering what you were saying about like going back to our K through 12 education. Like those are people who, like the youth that are now, going into college, like that age range, they've just come out of it, like they know what they got to relearn, like fresh in their minds. I love that. Yeah, and we have a question from Shannon Bennett who gave a talk, and she asks, how can the Academy incorporate Indigenous science in addressing climate change? And do you think an Indigenous scientist would want to work here? And how can we create a safe and welcoming and better space for Indigenous scientists here?
Yeah, so that's an interesting question, because I'm going to be the first Indigenous professor to teach Intro to Climate Change next year, right, in the University of Washington UW system. So I think that it's important for us to also not just teach Western science, but also Indigenous science, like use that place-based learning, education, work with examples that are locally, that are impacting Indigenous peoples, and seeing how they're already addressing climate change. Because oftentimes we forget that Indigenous peoples are addressing climate change as we speak. Like for instance, the tribes in the state of Washington where I reside, I'm on vacation right now, but like they're already leading the role or the movement to transition into renewable energy. Like you walk through reservations like the Tulalip tribe, they have wind turbines. And I think that seeing how Indigenous communities are already combating climate change will be a way to start addressing climate change, and building those small scale solutions that they're doing into larger scale solutions. And I think that Indigenous scientists want to work everywhere, right? And I think that oftentimes when we talk about those opportunities, oftentimes we question oh, is this really science? Why are we learning about Indigenous science or knowledge in a scientific presentation? That tends to be sometimes the question that comes up. And I think unraveling those questions and why we think a certain way, like why we think certain things are not science, I think will be another way to start, unlearning that process and then relearning for ourselves.
Yeah, I think that totally makes sense. And yeah, place-based learning definitely feels like something we can do, especially like, yeah, just considering how much amazing biodiversity and how many amazing local indigenous communities in the San Francisco Bay Area are, you know, the amazing work that they're doing as we speak, just like you said about Washington and those lands. But yeah, I think that's our time for questions. But thank you so much, Jessica. This was an absolute pleasure. And everybody who's in the comments and watching, definitely go pre-order her book. And I'll, you know, we'll link the website in the chat so you can go check that out. But yeah, thank you so much.
Yeah, Taguchi. Thank you.
And I think we're going to pop Lynn back on and do a little farewell.
Hi, it's getting a little loud behind me. But just want to say thank you to everyone watching tonight, and also a special thank you to Jack, Fayal, Dan, Shannon, and Jessica. Thank you so much for being a part of Night School tonight. Next month on October 7th, so there was just so much to share about kind of the human-nature relationship that we decided to do a part 2. So next month on October 7th, we'll be doing a part 2, but we'll be diving more specifically into forest fires and fire management, which is definitely an issue locally right now. And we'll also talk about the challenges we're facing considering fire history and climate change. So tune in next month.
Yes, indeed. And on that note, subscribe so you'll be notified right away, as usual, when we go live. And yeah, we'll see you all in roughly a month. Looking forward to it. Thanks again for tuning in.
Thanks, everyone.